Hi, this is Remembering the Past, the show where we talk about people who've died recently who've had a profound effect on our history, our society, or our culture. And in our ongoing series on Nobel Prize winners, tonight we're going to talk about Stanley Cohen, who died recently at the age of 97. Stanley Cohen was awarded one half of the Nobel Prize in Physiology or Medicine in 1986, along with Rita Levi-Montalcini, whose podcast we've done, and I refer you to it. They were awarded the Nobel Prize for their work in the discovery of growth factors. They first discovered an isolated nerve growth factor, NGF, and then Dr. Cohen went on to discover another growth factor, epidermal growth factor, EGF. NGF promotes nerve growth, and EGF promotes growth of other types of cells. This is how it's described in the official website of the Nobel Prize, nobelprize.org. Human beings develop from a single cell that divides to form new cells. These new cells then also further divide and multiply. Little by little, different types of cells with different functions are formed. After Rita Levi-Montalcini discovered a substance that promotes growth of the nervous system, in the mid-1950s, Stanley Cohen discovered another growth factor that promotes growth of cells in the skin and cornea. The discovery of what are now known as growth factors has provided a deeper understanding of medical problems like deformities, senile dementia, delayed wound healing, and tumor diseases. Stanley Cohen was a nice Jewish boy born in Brooklyn. He was a good student in school, and he went to James Madison High School in Sheepshead Bay. And let me digress here just for a second. 7 Nobel Prize winners have come from James Madison High School. 2 senators in the news these days came from James Madison, Chuck Schumer and Bernie Sanders. And Ruth Bader Ginsburg also came from James Madison. Carole King, Judge Judy, and Chris Rock went to James Madison. I'd be remiss if I didn't mention one other guy from James Madison, Roy Demeo, who was one of the most notorious hitmen for the mob. Stanley Cohen is the 6th person from James Madison we've done a podcast on, so I think they've moved into the lead in that respect. We've done Gary Becker, who was a Nobel Prize winner, David Frye, the comedian and impressionist, Martin Landau, the great actor, Mel Lazarus, the cartoonist, and Marvin Miller, who established the Major League Baseball Players Association. Anyway, Stanley Cohen grew up in a time after World War II when biochemistry was primarily the study of metabolic pathways. But about halfway into his career, Watson and Crick discovered the DNA helix, and that changed his whole field of research as it did so many others. Here's a little bit of his academic history from the introduction he was given in Sweden, not Norway, for his Nobel Prize speech.
Dr. Stanley Cohen received his training in chemistry and zoology at Brooklyn College and Oberlin College. After taking a PhD in biochemistry at the University of Michigan, 1948, he served as an instructor in the Department of Biochemistry and Pediatrics at the University of Colorado. In 1952, he moved to the Department of Zoology at Washington University in St. Louis, where he remained for 7 years until 1959, when he was recruited as a professor in biochemistry by the Department of Biochemistry in Vanderbilt University in Nashville. In 1976, he was named American Cancer Society Research Professor of Biochemistry at that school. Dr. Stanley Cohen's first contribution to growth factor research came in St. Louis, when acting as a collaborator in the research group of Rita Levi-Montalcini and Victor Hamburger. He successfully purified nerve growth factor. While testing the biological effects of NGF containing extracts, he made his discovery of the epidermal growth factor, EGF. After establishing the chemical nature and biological effects of both mouse and human EGF, he extended his leadership in the growth factor field by demonstrating the internalization of the EGF receptor complex and purification of the EGF receptor. His finding that the EGF receptor contained a domain with tyrosine kinase activity was a breakthrough in the growth factor field. From this discovery, a new concept emerged. Tyrosine kinase activity is a general step by which the action of growth factors is initiated. Dr. Rita Levi-Montalcini and Dr. Stanley Cohen were first to discover and isolate tissue growth factors. Their pioneering work has opened the research field of growth factors and has advanced our knowledge from a stage when growth and differentiation could only be described as phenomena to a situation when the role of the growth factors in cell proliferation, organ differentiation, and tumor transformation is generally accepted.
Well, as she said, Stanley Cohen did some of his Nobel work at Washington and St. Louis with Rita Levi-Montalcini. They were Hamish Jews. Rita Levi-Montalcini was an Italian Jew who was an émigré from Europe right during the war. She did the primary biology work on nerve growth factor, and Stanley Cohen did the primary chemistry work on it. His isolation of epidermal growth factor occurred when he moved to Vanderbilt. There have been 6 Nobel Prize winners associated with Vanderbilt, only 3 in physiology or medicine. One was Earl Sutherland, who we've talked about before. He was awarded the Nobel Prize in 1971. Max Delbruck was awarded the Nobel Prize in physiology or medicine in 1969. They have 3 others. One of them is Al Gore. He won it as a Nobel Peace Prize. I don't know where we put that one. Here Stanley Cohen discusses his discovery of epidermal growth factor in his Nobel laureate speech.
It's been about 25 years now since epidermal growth factor was discovered. As you all know, the phenomena consisted of taking extracts of salivary gland of mice as part of our nerve growth factor study. When we injected them into newborn mice, their eyes opened up earlier and their teeth came out earlier. The purified nerve growth factor did not have that effect. When I went to Vanderbilt, I decided to study that because I felt, you can be sure, not that it was connected to oncogenes, but that anything that disturbed the timing of a developmental process was worthy of study. So there was something in the extract that made eyes open up earlier. So using that eyelid opening assay as a bioassay, we were able to isolate the factor responsible for eyelid opening, and the molecule was sequenced in the old-fashioned way, and this is what it looks like. 53 amino acids, 3 disulfides, very stable, but most importantly, no lysines, only 1 amino group at the N-terminal, and anything you want to link up to the N-terminal doesn't matter. This molecule is biologically active. So that was very useful. Okay, so now we had a small protein molecule. If you inject it into a mouse, his eyes opened up.
It's now almost 50 years since Dr. Cohen discovered EGF. That speech was given 25 years after he discovered it. When the protein was discovered, it was a small protein, and initially they didn't know how it stimulated the eyes of mice and other animals to open, and they gradually found out in the next 25 years, in the interim between the discovery and that speech. EGF is a small protein, but it binds to a receptor on the cell membrane called the EGF receptor, and that small protein ultimately has major consequences because it causes cells to grow and also to divide. So one small 50-plus amino acid protein creates effects that even a layperson can ultimately see. At the time that Dr. Cohen gave that speech, they were only learning the details of how the EGF and the EGF receptor worked. They realized that when EGF and the receptor combine, they create a cascade of proteins inside the cell, and those proteins ultimately trigger the growth and division of cells. You heard the woman who introduced Dr. Cohen speak about tyrosine kinase. Tyrosine kinase was a part of the EGF receptor, and through its chemical composition, it acts as sort of a universal off-on switch inside the cell, and this is what she meant by the tyrosine kinase revolution. Since then, there have been a number of tyrosine kinase receptors discovered. There have been tyrosine kinase inhibitors discovered, and we realize that tyrosine kinase is a key factor in the development of many human diseases. So tyrosine kinases, receptor and non-receptors, are overexpressed in many solid tumors, and a whole new field of oncology treatment was based on this fact. So you saw many new drugs developed once they realized the tyrosine kinase receptor mechanism and its relationship to cancer. There's also a special type of tyrosine kinase protein that's found in the central nervous system of Alzheimer's patients, but no one is sure yet what the significance of that is. And people have started looking at it as a potential target for the treatment of Alzheimer's disease. In fact, there are a number of diseases now where tyrosine kinase inhibitors are standard treatment, and they were unheard of at the time that Dr. Cohen gave that speech in 1986. So you can see that the work of Dr. Cohen and Dr. Levi-Montalcini was instrumental in opening up an entire new branch of medicine. One of Dr. Cohen's mentees at Vanderbilt, Dr. Tom Daniel, said that in view of the therapeutics that target the EGF receptor to treat lung, colon, and head and neck cancers, of all biomedical science, the EGF story has been legendary. Dr. Daniel started a fund, the Stanley Cohen Innovation Fund, to bring increased attention to his legacy of innovative research. It's important not just for Vanderbilt, but also for the world, for catalytic innovative research to be done in Stan's name. We have an opportunity to recognize the impact of Stan's work and to heighten awareness of it. He really changed the landscape, not just for Vanderbilt, but for patients and certainly for the entire scientific and medical community. Carlos Arteaga, who's a professor of breast cancer research and director of the Center for Cancer-Targeted Therapies at Vanderbilt, said that Dr. Cohen opened a field of biology that allowed me to focus the most critical part of my career at the beginning on ways to interfere with signaling by the family of EGF receptors in cancers that are dependent on these molecules. Another one of his colleagues, Graham Carpenter, said that Dr. Cohen had a fantastic ability to focus, in this case on EGF and nothing else. He was probably the best experimentalist I've ever met. He spent an immense amount of time just thinking about how to test a hypothesis. That thinking often happened as Cohen walked the hallway smoking a pipe, or after smoking was prohibited, carrying a pipe in his mouth. The pipe was a thinking tool that usually got left behind when he stopped for conversations. Carpenter said he had a whole box of pipes in his office, and the pipes ended up all over the medical center north. He had a very relaxed way about him, the way he dressed, the way he entered into casual conversations, but there was a lot of intense stuff going on inside him. Well, I'm going to close on that note. I want to thank my producer and IT genius, Sid Tepps. As I said, Stanley Cohen came from Brooklyn. He went to Brooklyn College, but once he graduated from Brooklyn College, the interesting thing is, like many others, he pretty much left Brooklyn. He spent most of his time in the Midwest, West, or South, Michigan, Colorado, St. Louis, and Nashville. And he became another one of the countless kids from Brooklyn who went on in his specific field to change the world. So as a final tribute to him, we're going to play a Brooklyn song. We've already played Brooklyn Roads by Neil Diamond, my favorite, so we'll play another good Brooklyn song about someone leaving Brooklyn and not returning. And this one's by Gene Pitney from 1965, The Last Chance to Turn Around. Here it is to honor Dr. Stanley Cohen.
Gonna keep these wheels of mine hovering round on the last station to Brooklyn, the last chance to turn around.